Hello students. Now as you can see, this picture summarizes the process of photosynthesis, a process that happens in plants for them to produce their own energy. And when we speak about energy, we are speaking about glucose, sugar. Now, before I start explaining about the photosynthesis, I need you to understand one thing, that this PowerPoint include a lot of pictures and less explanation. So I need you to pay attention. You may also use your books or your lecture or the slides from Dr. Juwair or Ms. Raghda, the one that they're providing, to create your own lab report, because after all, you will have to write a lab report for photosynthesis. So to define photosynthesis in very simple words, it's a process in which plants produces their own energy. This is a summary. They need a certain intakes, sunlight, carbon dioxide, water, for the process to happen, and the outcomes are glucose and oxygen, as you can see over here. So basically this is a summarized for the process of photosynthesis. Our purpose today in our experiment, we will experiment the importance of sunlight. Since we said that the sunlight is needed for that process, I will use I will take the sunlight as my experiment today, as a purpose of my experiment. So I will discuss now what we need for the materials. Of course, we need sunlight. We cannot grab a sunlight, but we need leaves for our experiment. And basically, why leaves? Because the chloroplast occurs in leaves, mainly for the process of photosynthesis. So this is a chloroplast, and this is what happens inside it. Okay? So before I start with the materials and learning about the materials, I need to revise again. The purpose is experimenting the importance of sunlight in the process of photosynthesis, since it is one of the most important intakes that is needed for the photosynthesis. What is photosynthesis? It is a process in which plant produces their own energy or their own food or their own glucose. It's the same. Okay, this is the intake that we're looking for. All right, so basically we defined photosynthesis and we learned our purpose, and from that you can create your own question and hypothesis. So I'm discussing what we need as materials. So far, as you can see, this is a Clifton. I use it as a hot plate to heat the water. This is a beaker filled with water. So first of all, we need a Clifton hot plate, beaker filled with water, and another beaker over here filled with methanol, which is an alcohol. Okay, I need 2 leaves. One was kept facing the sun and the other leaf is kept away from the sun. I kept it in a drawer for minimum 48 hours, and I kept it in a nylon bag so it won't dry. I need forceps, iodine, and my pipette. And this is what we call it as petri dish, because I will apply the leaves after I'm done from boiling it and apply the iodine in the petri dish. Okay, so this is basically what we need. This is the petri dish for the leaf facing the sun, and this is the petri dish for the one that was kept away from the sun after boiling them. Now, why do I need to boil the leaves in water? It is very important to understand why am I using these materials. It's very common sense. When I boil the leaves in water, I will break the cell wall that surrounds the cells. Now we learned that plant cells has cell wall around the cell to protect it. So we need to break it in order to go inside the cell and look for the glucose, not glucose, basically the starch, because after they consume the sugar that is produced from photosynthesis, the excess sugar will be stored inside the leaves in the form of starch, not glucose, in the form of starch. So I need to look for the starch inside the cell, so I will break the cells, the cell wall, by boiling it in hot or boiling water, and then I will boil methanol. Now I know there is no any differences, but this is methanol, this is alcohol. The reason I will boil it the second time with alcohol to bleach the green pigmentation, because I need to look for the starch. Starch will be colored and reacted with the iodine. Remember lab number 1, it reacts with the iodine. It will give me a black bluish coloration, but because of the green color I won't be able to recognize it. So I will bleach out the green pigmentation by boiling it in methanol. Now, after boiling the leaves in water and methanol, okay, I will simply apply the iodine and look for the color change. So basically 3 main steps: boiling water, boiling methanol, then apply iodine. The reason I'm boiling it in water to break the cell wall, I boil it in methanol to break, I boil it in methanol to clear the pigmentation or bleach the green pigment, and then I will apply iodine to look for the black bluish coloration, which means if a black blue coloration appears on the leaves, this is a starch. So this means that there is a photosynthesis and it created sugar and it consumed it and stored in the form of starch. If it stays without any black coloration, stays yellowish, stays leaf as it is, it means there is no starch. And by meaning no starch, it didn't do any process of photosynthesis because this was kept away from the sun. So this is why I told you we are experimenting the importance of sunlight. So I told you now the answer, but we will wait after we do the experiment, okay? Now it's been almost 6 minutes since I added in a boiling water, and I'm not sure if you can see that on the leaves over here. I'm not sure if you can just, this is hard for me, I cannot hold it, but I will just explain it. Over here you can just focus, you will find a lot of air bubbles on the leaves. That air bubbles indicate that the cell wall is broken, carbon dioxide is released outside the leaves. This is my indication. I don't want to grab it from up, from the water, sorry. I actually added here the leaf that was kept away from the sun. And this is the leaf that was kept facing the sun. I will keep it for 4 minutes to complete the whole 10, and then I will show it to you in a boiling methanol. Now I'm removing the leaves from the water and I will apply it in the methanol, and I will wait for 7 minutes. So this is after 7 minutes of boiling the leaves in the methanol. And as I said, the reason we are boiling it in methanol to bleach out the green pigment. Look how the leaf becomes yellowish in color. I will apply it in my petri dish. Over here, and I will apply iodine on top of it. So here I am applying oh, I need you to see this. Can you see the black dots that forming? Of course, if I keep it more time, look at the black spots. If I keep it more time, the whole leaf will turn into a black, which means this is starch, and meaning that there is a starch on the leaves. Look, becomes dark in color. The starch means that there was photosynthesis happen in the leaves. Glucose was formed and consumed, and this is the excess starch was stored inside the leaves. Look how beautiful. And I will just keep it, and I will take a picture of the result in a minute. Now for the one that was kept away, see, nothing, stays yellow, nothing happened. Okay, so this is the experiment for today, photosynthesis. Look how starch was, the black coloration was filled all over the leaves because it was facing the sun, while the leaf that was kept away, nothing happens to it, stays yellowish as it is. Okay.